Welcome everyone to the Genetic Engineering and Biotechnology News webinar series. Our presentation today is entitled Reliable Binding Kinetics and Affinity Determination Using Biolayer Interferometry. Hear from the experts. I'm Jeff Bogaliskis, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. Binding kinetics are typically a key determinant that scientists rely on during the screening and selection process of lead therapeutic monoclonal antibodies. Numerous factors can potentially influence accurate measurement of binding rate constants when using biolayer interferometry, or BLI, platforms. Advances in biosensor technology over the past several years have addressed the growing demand for more accurate characterization of biomolecular interactions, thus allowing BLI to surpass surface plasmon resonance as the go-to method for binding kinetic measurements. Let's meet our panel so they can outline some recent data for the measurement of binding kinetics and affinity determination, as well as provide guidance on how updated BLI methods can assist your laboratory workflow. Our first speaker today is Dominic Andrada, Senior Product Manager at Forte Bio, a division of Paul Life Sciences. Mr. Andrada will provide a brief introduction of BLI technology and how it can be applied to streamline your current research. The second presentation will be given by Vishal Kamat, scientist at Regeneron Pharmaceuticals. Dr. Kamat will discuss tips and tricks for optimizing the binding kinetic analysis on the Octet HTX system and share observations on the influence of various assay parameters for characterizing antibodies across surface plasmon resonance and BLI platforms. Our final presentation will be provided by Michael Brown, scientist at Adimab. Mr. Brown will review picomolar binding affinity data measured by the BLI ELISA technique on the Octet system. Before our speakers get started, I want to encourage the audience to submit questions for our Q&A session at the end of the presentations. We'll try and answer as many questions as we can, so simply type the question into the Q&A box on the left-hand side of your screen and hit submit. So without any further ado, let's begin our webinar. Dominic, take it away.
Thank you, Jeffrey. Good morning, everyone. I'm Dominic Andrada, Senior Product Manager at Forte Bio. It's good to be here with you this morning. I'll start the presentations with a short talk on biolayer interferometry, also known as BLI, and how BLI can streamline your research workflow for large molecule drug discovery. Biolayer interferometry, or BLI, is a label-free method to analyze protein interactions in real time. It's an optical method measuring changes in molecular thickness on the biosensor surface as a mobilized ligand interacts with its binding partner or analyte in solution. Every biosensor is composed of a hub and a fiber optic coated with an optical layer, a biocompatible matrix, and finally coated with the desired surface chemistry. BLI detection starts out by measuring the interference pattern of a given biosensor. White light is directed down the fiber optic, and reflected signal coming from both the optical layer and the tip surface generate an interference pattern. Relative intensity is measured across multiple wavelengths. This illustration shows the tip of a biosensor on the left-hand side and a reference interference pattern in gray, which would correspond to a baseline if no analyte were bound to the biosensor. When the biosensors are dipped into sample wells containing target molecule, binding occurs on the tip surface with a specific immobilized chemistry or ligand. This interaction increases the number of molecules bound, increasing the molecular thickness on the tip surface, and causes a change in the interference pattern. The new interference pattern is illustrated as the red trace on the right side of this slide. Changes in the interference pattern, or delta lambda, are monitored in real time. The delta lambda for this illustration is a change from the gray to the red interference patterns. Looking at the interference pattern a little more closely, the change in relative intensity is a function of both the change in distance from between the two reflecting surfaces on the biosensor tip and the change in the phase shift of the wavelength being monitored. This relationship is used to plot nanometer shift as analyte associates, then dissociates from its ligand on the biosensor tip. The resulting sensogram is nanometer shift, or nanometer, on the y-axis and the seconds on the x-axis. Quantitation assays monitor the binding rate of the target protein to the biosensor tip, whereas kinetic assays show both the association step and dissociation step. Of course, the quality of interactions is due in part to the surface chemistries on the biosensor surface. And Forte Bio has the broadest menu of biosensor chemistries for label-free detection of quantitation and kinetic assays. This provides for more flexible assay optimization for optimal results. Another benefit of these sensors is not being constrained by mandatory regeneration. Along with the large menu of biosensors, we have other tools to support your work. Here are some examples of recent application notes that you can find on our website. Listening to our users, we have found increasing interest for looking at Fc gamma receptor interactions using Octet, and the ease of use and high throughput nature of the HTX has made it a favorite for people working on epitope binning. More resources for different applications can be found on our website. The 4 value statements on the bottom of this slide are some of the reasons BLI is better able to address evolving customer needs that other platforms are not addressing. Reliable kinetic analysis, handles your toughest samples, fast protein quantitation, and throughput when you need it. The complete lineup of our Octet systems is shown above, with our flagship instrument, the Octet HTX, in the rear center. The recently launched Octet K2 is shown in the front right. Next to it is our manual Blitz system. The next 2 speakers will share how they use their octets for their work on reliable binding kinetics and affinity determination.
Thanks, Dominic. That was a great presentation to get us started, and you provided us with a nice introduction on the specifics of the BLI technology, so we thank you. Before we move on to Vishal's presentation, I want to remind everyone once again to submit questions for our Q&A session at the end of the presentations. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. All of our speakers will be available to answer your questions after the final presentation. So, without further ado, Vishal, the floor, as they say, is now yours.
Thanks, Jeff, and good morning to everyone. I would like to thank Forte Bio for giving me this opportunity to share my experience in designing binding kinetics assay on the Octet platform. In today's presentation, I will provide some insights and few tips and tricks in designing binding kinetics assay on Octet. BLI has been a relatively new technology compared to the well-established SPR biosensor platform, and there is a growing interest in the label-free community to use this technology in order to design binding kinetics assay in an effort to measure reliable rate constants of protein-protein interactions. In today's presentation, I will provide a brief introduction of binding kinetics assay, the definition of binding rate constants such as the association and dissociation rates. I will also provide a brief comparison between SPR and BLI technology platforms. But the major part of my presentation will focus on various factors that can potentially influence the measurement of binding kinetic parameters and different steps required to optimize experimental conditions, such as optimization of buffer conditions, selection of appropriate well plates, antibody surface density, and steps involved to achieve reproducible map capture level. Later on, I will briefly talk about designing titration kinetics assay and single cycle kinetics assay on the Octet platform. Finally, I will conclude my presentation by showing a compatibility study between Biacore and Octet platforms involving measurement of binding rate constants for a total of 160 unique antibody-antigen interactions binding to 11 different molecular weight antigens. So any chemical reaction that we have been taught right from our undergrad is when you have two reactants A and B, the complex formation, which is AB, is entirely governed by two parameters. The K-on, which is the forward reaction rate or the on rate, which is dependent on the concentrations of the reactants A and B, and the K-off, which is the backward reaction rate, and which depends on the inherent property of the reactant to go back for the product to go back to the reactants. The rate of a chemical reaction is provided below towards the end of the slide, which is dependent on the concentrations of the reactants A and B times the on rate and minus the A concentration of the product AB times the off rate. So in a typical label-free binding assay, the K-on is what stands for the Ka, or the association rate constant. The K-off stands for Kd, which is the dissociation rate constant. So the entire equation of a chemical reaction in the case of a biomolecular interaction is as shown in this particular slide. So if we consider B as the antibody, in this particular case the ligand which is on the surface, and B which is the analyte which is injected over the ligand surface, then AB is the complex formation which we observe in a particular binding response in the case of a real-time binding platform. Here is an example of a typical real-time binding data performed by injecting multiple concentrations of analyte over the ligand surface. A typical binding data comprises of two phases: the association phase, wherein we inject the analyte over the ligand surface, and the dissociation phase, wherein we allow the dissociation of the bound analyte to fall or dissociate from the ligand surface. During the dissociation phase, we do not inject any analyte over the ligand surface, and any change in the observed binding response is due to the inherent property of the analyte to dissociate from the ligand. This rate of change is governed by the dissociation rate constant, the Kd, of that particular interaction. On the other hand, during the association phase, you have two events occurring simultaneously, which is the binding of the analyte to the ligand and also the dissociation of the bound analyte from the ligand. So during fitting the real-time binding data, we first need to calculate the dissociation rate constant, followed by the fitting of the association phase to measure the association rate constant. And finally, we measure the binding affinity, Kd, of the interaction by calculating the ratio of Kd over Ka. As everyone's aware, surface plasmon resonance is the basic working principle of different label-free platforms such as Biacore, Proteon, SensiQ, MAS1, and MAS1 from Sierra sensors. These biosensor platforms rely on the resonate oscillation of the valency electrons on the surface of a thin gold film following the stimulation of the incident light. On the other hand, BLI, which is the working principle of the Octet platform, relies on the interference of light. However, irrespective of the differences in the working principle of SPR or BLI biosensor platforms, we always observe real-time binding curves as shown in the figure, containing the association phase, wherein we inject the analyte over the ligand surface, and the dissociation phase, wherein we study the dissociation of the bound analyte from the ligand surface. But there are certain differences between SPR and BLI platforms. SPR measures change in the mass, and the binding response is referred to as RU, which stands for resonance units, whereas BLI measures change in the surface density, and the binding response is measured as nanometer shift. Both SPR and BLI platforms can adopt capture method, wherein the ligand is non-covalently captured on the sensor surface, or the direct method, wherein the ligand is covalently immobilized onto the sensor surface using different surface chemistries. SPR is a continuous microfluidic device, and samples are injected into the IFC at a particular flow rate defined by microliters per minute. On the other hand, in the BLI platform, samples are dispensed into the well plate, which is placed on top of an orbital shaker. There are different additives in the sample buffer, such as salt, detergent, which can affect binding interactions for both SPR and the BLI platform. However, in the case of the BLI, we need to add BSA in the sample buffer, and I will talk more about the importance of BSA in the subsequent slides. Both the platforms demonstrate challenges such as characterization of mass transport limited interactions. In addition to this, there are several other factors that can influence a typical binding interaction, such as the dextran effect observed in the case of the SPR platform, while the BLI platform, which has a planar surface, researchers have reported to experience rebinding of the analyte to the ligand surface. In order to support the high throughput requirement of Regeneron Pharmaceuticals, we have a whole bunch of different technology platforms available in the HTS Biomolecular Center. We have four different SPR instruments from Biacore. We also have the MAS1, which is the high throughput instrument from Sierra Sensors. We also have the Octet Red 96 and Octet QK, along with the Octet STX from Forte Bio. In order to measure solution-based affinities, we have the Kinexa 3200 with the auto sampler, and we also have the Microcal Auto ITC 200. Apart from that, we also have a whole collection of Luminex instruments, which is essentially used during the primary screening. For today's presentation, I will just focus on the use of Octet STX in designing binding kinetics assay. Before developing any binding kinetics assay on the Octet platform, we first aimed to optimize sample buffer condition by studying the effect of different additives on the baseline stability of the captured antibody. Since all the antibodies developed by Regeneron Pharmaceuticals are fully human, we used AHC sensors to capture the antibody in this particular study. To check for sensor-to-sensor variability, 4 AHC sensors were used to capture fully human antibody prepared at the concentration of 2 micrograms per ml. EPS buffer containing different concentrations of BSA was used in this particular study, and the concentrations of BSA varied from 0 mg per ml all the way to 1 mg per ml. Antibody capture time, or the loading time, was fixed at 4 minutes, and we studied the baseline stability of the captured antibody for 1 hour. Here is the data of the binding study we performed for the effect of BSA. We did not see any significant difference in the map capture level for samples prepared at 1 mg per ml BSA or 0.1 mg per ml BSA. However, if we reduce the concentration of BSA to 0.04 to all the way till no in the absence of BSA, we see a dramatic change in the capture level. Moreover, we also see a fast dissociation of the captured antibody, suggesting that the capture or the stability of the captured antibody is significantly compromised in the case of reduced to no BSA conditions. After further looking into the baseline stability of the captured antibody, we found that there is a lot of sensor-to-sensor variability even if we add 1 mg per ml BSA. And we initially see an upward drift in the captured antibody, and then sometimes the rate of dissociation is much faster for certain sensors, as observed in the case of 0.4 mg per ml BSA. Collectively, this data suggests that addition of BSA is just not enough to achieve stable baseline stability of the captured antibody. We further looked into the addition of detergent, and turns out that when we supplement the sample buffer with 1 mg per ml BSA in addition to 0.05% WIN20, we do achieve a stable baseline, or the captured antibody stays bound to the AHC sensors. So for all the subsequent studies, we have been using 1 mg per ml BSA containing 0.05% of tween20 for all the binding assays. As I mentioned earlier, in the case of Octet platform, all the samples are dispensed either in a 96 well plate or on a 384 well plate, and these plates are placed on top of an orbital shaker. Moreover, there are two types of 384 well plates. We have a flat bottom well plate and we also have a tilted bottom well plate. Having multiple options of plate types, it can be really puzzling to select appropriate plates which is better suited to perform binding kinetics assay. So we performed a simple plate test on the Octet HDX. We used an AHC sensor and then dipped into different wells containing just the sample buffer. And in this particular case, we used 1 mg per ml BSA and 0.05% tween20 within a heap yeast buffer. Here is an animation of the experimental design. So we selected, in order to achieve sensor-to-sensor variability, we used 4 AHC sensors and they were dipped in different wells containing the sample buffer. Here is the data for a 384 flat bottom well plate. For most of these wells that the sensors are dipped in, we did not see any bulk shift. However, we did see some small amount of bulk shift when the sensors were dipped in wells A21 and A22. Moreover, we also saw an edge effect when the sensors were dipped in location A24. On the other hand, when we used a 384 tilted well plate, we did not see a significant bulk shift when the sensors were dipped in locations A17 all the way till A23. However, we continued to see the edge effect. So if you have your samples, you would want to keep your samples dispensed in any well except for the last well location, which is A24, for performing binding assays. Bulk shift was found to be even more dramatic when we used the 96 well plate on the Octet HDX platform. And apart from seeing a gradual drift in the baseline, there was a lot of sensor-to-sensor variability observed, which could be because of the surface artifacts which gets reflected from the bottom of the 96 well plate. So when we moved the plate and used an Octet Red 96 instead, which has a tilted platform where the plates are placed, we did see a significantly reduced bulk shift, and we did not see any drift in the baseline either. So collectively all the data suggest that in order to design kinetics assay on the Octet platform, you either need to use a 384 tilted well plate or used an instrument which has a tilted platform, as in the case of the Octet Red 96. Quite a few published literatures are currently available on the effect of surface density on binding kinetics, and there is a general consensus that reduced surface density is highly recommended to perform high-resolution kinetics assay. But most of these studies are performed on SPR platform, and there is very limited information currently available on the Octet platform, which provided the rationale of this particular study. We used AHC sensors to capture fully human monoclonal antibodies, and in this particular case, we selected 8 antibodies per antigen for a total of 4 different molecular weight antigens, with molecular weight ranging from 20 kD all the way to 68 kD. We used the 16-channel high-sensitivity mode on the Octet HDX platform. Based on the in-house SPR data, appropriate single antigen concentration was selected to perform this particular study. Binding rate constants measured on the Octet HDX was compared with those generated using Biacore by calculating the correlation factor, which is essentially the ratio of the kinetic parameters measured using Biacore to the Octet. One of the important things that we did in this particular study, we used single reference subtraction to subtract out the baseline drift observed for the captured antibody. Here is the data that we generated for in this particular study. We have 8 different antibodies labeled in different colors and different shapes. And for a 20 kD antigen, we did not see a significant difference between the measured rate constants as a function of surface density. Shown in serrated lines suggests that the correlation factor is within 3, which essentially indicates that the rate constants are within 3-fold of each other. And in all this particular study, we did not see a big difference, and all the values were within 3-fold for the values measured on the Octet versus the Biacore. We did see a slight increase in the correlation factor for lower density surfaces, especially in the case of the binding affinity, but binding surface density of 0.6 to 0.7 seems to be working, and the rate constants were within 3-fold of each other. However, in the case of a 28 kD antigen, we did observe a slight increase in the measured rate constant of the Biacore instrument. The correlation factor, which is greater than 3, suggests that the rate constant, or the association rate constant measured on the Octet, turns out to be slower for a higher density compared to the antibody surface density of 0.6. When we extended the study for 48 kD antigen and 68 kD antigen, we did not see any dramatic change in the measured rate constants compared to what we saw earlier with the 28 kD antigen. And all the rate constants were within 3-fold of each other between the Octet and the Biocore platform. So based on this study, we would recommend to capture around 0.6 to 0.7 nanometer of the antibody for characterizing a wide range of binding affinities of the antibody. Here is an example of the curve that we generated in this particular study. Shown here are representative figures for the antibody binding to the 28 kD antigen, for the antibody binding to the 28 kD antigen, 48 kD antigen, and the 68 kD antigen. It has been previously reported that the Octet platforms do have an issue, which is the rebinding of the analyte to the ligand surface. However, if you focus on the binding curve shown under the blue box, we did see dissociation of analyte from the ligand all the way to the baseline, suggesting that there was no rebinding of the analyte to the ligand surface. Likewise, the 28 kD antigen shown in the blue box also dissociates all the way to the baseline. So collectively, we believe that if we optimize the appropriate buffer conditions and also appropriate antibody surface density or the ligand surface density, you can overcome the rebinding effect which has been previously reported. There is a general concern amongst the label-free community that Octet biosensors exhibit a lot of sensor-to-sensor variability, and it also fails to achieve similar capture level. While performing kinetics assay, it is important to achieve similar map surface densities so that the observed binding data can be globally fit using one-to-one binding model. Here is an example of map capture step using 8 different AHC sensors. These sensors were dipped in antibody sample prepared at 20 microgram per ml and dispensed into 8 different wells with 120 seconds of loading step. As highlighted in the red box, we observe varying capture level for different sensors. However, if we just focus on the first 20 to 30 seconds of the capture step, we do not see any significant variability in the map capture level. Moreover, based on the previous study on the effect of map surface density, we typically aim at capturing 0.6 nanometer of the antibody, which can be easily achieved by shortening this loading step. Here is an example wherein 8 different AHC sensors were dipped in the same antibody samples shown in the previous slide, and following 12-second loading step, we observed minimal sensor-to-sensor variability, and we achieved similar antibody capture levels for all the 8 sensors which were dipped in these antibody samples. Here is another example wherein instead of using 20 micrograms per ml of the antibody, we prepared antibody samples at 4 micrograms per ml and loading step of 120 seconds. Even in this particular case, we do see sensor-to-sensor variability. However, like as observed in the previous slide, there is minimal variability if we reduce the loading time to about 50 seconds. And here is an example wherein the same antibody samples, where we have different AHC sensors dipped into the same antibody sample, and we see very minimal sensor-to-sensor variability, and we achieve similar map capture levels within this particular time period. So if you want to achieve similar capture level, we would recommend to either use 20 micrograms per mil of the antibody for 8 to 10 second capture step or loading step, or you can also do 4 micrograms per mil of the antibody with 50 seconds of the loading step, and you would see very minimal variability from sensor to sensor. Once we gained enough confidence on optimal experimental conditions required to design reliable binding kinetics assay, we expanded our study in designing titration kinetics and single kinetics assay. To achieve this, we selected 5 different molecular weight antigens, and we have 3 antibodies per antigen, making it a total of 15 different antibody-antigen interactions. For the titration kinetics assay, we used 4 to 5 different concentrations of the analyte, and for the single cycle, we used 4 concentrations of the analyte. And all the analytes were diluted by 3-fold in the kinetics buffer, which is essentially Heapy's buffer containing EDTA and tween-20 and 1 mg per mil BSA, and we also had sodium azide in our sample buffer. The plate shake speed is maintained at 1000 RPM. Here is the data that was generated on the Biacore 4000 for a 14 kD antibody binding to 14 kD antigen. And all the sensors shown in here have duplicate concentrations for the same analyte. When you compare the Biacore data with the Octet, the data generated from the Octet HTX, we do not see any significant change or difference between the shape of the curve or the kinetics profile of the antibody, be it the single cycle kinetics, which is shown in the right panel, or the titration kinetics shown in the center panel of this particular slide. Even in the case of 29 kD molecular weight antigen, we did not see any significant difference in the shape between the data generated on the Biacore 4000 versus the Octet HTX. Here is another data set wherein the data for the antibodies binding to a 48 kD antigen, and we did see similar result, no significant difference in the binding kinetics or the binding profile of the antibody to the antigen. So collectively, the 5 different molecular weight antigens shown in this particular slide, if you compare single cycle kinetics versus the titration kinetics that was generated on the Octet HTX platform, we did not see any significant difference in the measured rate constants. Shown in the red serrated line that indicates 3-fold difference, and all the values were within 3-fold of each other. So with the Octet HTX platform, you can perform serial titration kinetics assay, but you can also perform single cycle kinetics. What was also promising was if you compare the kinetic rate constants measured on the HTX versus the Biacore 4000, and in this particular case, it is the titration kinetics assay, most of the antibodies or most of the measured rate constants were within 3-fold of each other. We did find one antibody antigen interaction in the case of the binding affinity was outside the 3-fold difference, but that could just be a subtle difference between the type of the sensors versus the dextrine matrix versus the planar surface on the Octet. But overall, most of the binding rate constants were within 3-fold of each other. After gaining enough confidence in optimal conditions required for designing kinetics assay, we further expanded our study and we thought of including 160 different antibody panel, which comprises of antibodies binding to 11 different molecular weights with molecular weights ranging from 14 kD all the way to 105 kD. There was one small difference between this particular study versus the other. we did use regeneration step to reduce the cost of this particular experiment. So in this particular case, we tried different conditions, and turns out if you use 20-second incubation in 20 millimolar phosphoric acid, and we do it 3 times, we are able to completely regenerate the captured antibody from AHC sensors. And one of the other important thing of this particular study is that all the experiments were performed in triplicates as 3 independent experiments. Here is the experimental setup for this particular screen. We precondition the sensors using an initial regeneration step. And as mentioned in the previous slide, we prefer to use a short capture step, so all the antibodies are prepared at 20 micrograms per mil, and we're using 8-second capture step in this particular study. We also found that if you use a dummy capture step before the start of the experiment, it helps in reproducible capture level of the antibody following the subsequent regenerations. And here is where we have our experimental map capture step. Here is the data when we open it in the data analysis software, and we export the entire raw data into a text file. Later on, we import the entire text file into the scrubber file, which we use for data analysis. Shown here, we have the map capture step and the binding of different analyte over the antibody surface. So when we crop and align the entire data set, here is how the raw data looks like before single reference subtraction. And here is the buffer step for the entire experiment. One of the things we did observe during the regeneration step is there is a lot of baseline drift observed when you regenerate the sensors. And there is a lot of sensor-to-sensor variability in this baseline drift. Here is the process data for different molecular weight antigens from 97 kD and 38 kD, and all the concentrations presented in this particular figure are in duplicates. So it is encouraging to see that all the binding curves do overlap one over the other for duplicate concentrations. Here is the data set, and these are all representative figures for the entire panel of antibodies that were used in this particular study. And in this case, I'm showing the binding curve for 32 kD antigen all the way down to 14 kD antigen. So collectively, in this huge benchmark study, for the entire 11 different molecular weight antigens, most of the values were within 3-fold of each other, and shown in here is the association rate constant comparison between the Biacore 4000 versus the Octet STX. Shown here is the KD, the dissociation rate constant between the 2 platforms, and then finally the binding affinity that was calculated between the 2 platforms. We did observe that certain molecules or certain rate constants were more than 3-fold of each other. But the source of all this discrepancy is due to the baseline drift observed during the regeneration step. So it works okay if you're into the screening mode, but if you're interested in generating high resolution binding kinetics data, then I would not recommend to regenerate the sensors. So in conclusion, for designing any binding kinetics assay, it's extremely important to add 1 mg per mil BSA and with 0.05% tween 20 in your sample buffer, and that which enables to have a stable baseline of the map capture. You either use a 384 tilted well plate, which is compatible on the Octet STX platform, the Red 384 and the QK 384, or you would prefer to use a tilted platform on the Red 96, just to avoid the bulk shift observed. Map surface density is extremely important, and I did demonstrate few cases wherein overloading the sensor results in lower measured association rates compared to the Biacore instrument. But it's extremely critical when you have a very high affinity interaction that needs to be characterized. So you might want to further optimize the conditions, especially when the on rate of a particular interaction is greater than 10 to the power 6. You can achieve reproducible map capture level using the AHC sensors, and we have two different strategies. Either use short capture step with 20 micrograms per mil or a 50-second capture step for 4 micrograms per mil of the antibodies. The single cycle kinetics can be performed on the Octet HDX, and the measured rate constants were pretty comparable with the titration kinetics on the HDX and also with the Biacore 4000. And finally, the binding rate constants measured on the Octet HDX platform was found to be compatible with the Biacore 4000. We did observe baseline drift during the regeneration of the AHC sensors, so as I mentioned earlier, you should probably avoid regeneration for high-resolution kinetic assay on the Octet instrument. With this, I would conclude my slide, and I would like to acknowledge our Senior VP, Nicholas Papadopoulos, VP of Research and Development, William Olson, along with our Senior Directors, Tammy Wong and Joel Martin, for their continued support and inspiration. I'm also grateful for Ashik Rafiq for supporting me and for giving me enough time and freedom to develop kinetics assay on the Octet platform. With this, I would like to conclude my presentation. Thank you very much.
That was great. Thanks very much, Vishal. I think our audience now has a great sense of the power and versatility of BLI. Plus, you provided them with some helpful optimization tips, so we thank you very much for that. It looks like we're already starting to get some really great questions rolling in, so I encourage the audience to keep them coming. All right, so why don't we move along and begin our final presentation of our webinar. Mike, when you're ready, you have our attention.
Thanks, Jeff. And thank you also to the GEN webinar organizers for the opportunity to speak on behalf of my colleagues here at Adimab. Today I will go over a solution equilibrium titration assay we developed that combines Forte Biolayer interferometry and colorimetric ELISA detection reagents. I will then show how we use this assay to measure low picomolar antibody antigen affinities in a high throughput manner. Before I talk about BLI ELISA, I would first like to tell you a little bit about Adimab. We are the industry's premier antibody discovery platform. We have founders that have been successful in both academics as well as industry. We have licensed our technology to 3 large biopharmaceutical companies and continue to look for more partners in this area. And we're based in Lebanon, New Hampshire, and also have a small computational biology group in Palo Alto, California. So Adimab's an early development discovery company, and we have nestled ourselves between target identification and basic research and preclinical development. We've partnered with over 50 different companies to develop antibodies and bispecifics for multiple targets, and we have no desire at all to develop our own drug molecules. We're really focused solely on providing a diverse set of therapeutic candidates to our partners. So what sets us apart from other antibody discovery companies is quality, speed of delivery, and IgG diversity. We can complete a discovery cycle in about a month's time, and it all starts with a partner providing antigen and applying our proprietary IgG library and yeast display technology to find binders with a partner-specified phenotype. Once identified, we have the antibody sequenced, scaled up, and purified so that they can then run through our characterization assays, which include purity determination, binding kinetics, and a series of antibody developability assays that we've designed and developed here at Adimab. If the materials meet the criteria for delivery, we'll deliver them to the partner. If not, we can affinity mature or pressure in a different manner to guide the selections to a more desired set of antibodies, and then push it through the process again until we finally have antibodies that actually meet the criteria of both the both the partner as well as our own selection criteria. So enough about Adimab. Let's talk about measuring low picomolar affinities. There are 2 typical methods of choice. One way is to measure the association and dissociation rates and then calculate the KD. Vishal gave a great introduction on that. The so-called kinetic experiment. These assays are typically performed on SPR or BLI instruments, but as I'll show on the next slide or 2, they do provide some challenges when you're looking at low picomolar interactors. What I plan to focus on today are solution equilibrium methods, and in particular on high throughput solution-based methods like MSD shown here, and the assay I plan to talk about in much greater detail, the Biolayer. So when working with high affinity interactions, we're often dealing with systems that have very slow dissociation rates. The rule of thumb for measuring accurate off rates is that we would like to observe at least a 10% signal decay for the dissociation. This means we need to wait at least 2 hours and sometimes more to obtain accurate off rates. Waiting this long can introduce baseline drift, which can lead to inaccurate measurements. In some instances, the association rate can also be too fast to be measured. This has led us to look for a solution-based method for measuring these high affinity interactions. So Kinetics is the gold standard for measuring high affinity interactions via a solution-based method, but it was also too slow for our throughput purposes. This figure shows that you have multiple steps that are necessary in order to measure a single titration data point. First, you have to pack your bead column. Then you have to come in with your mixture of antibody and antigen. You have to flow that back out. Then you have to come in with your label, flow that in, flow that back out. This all takes time, and time is really money for us. And so it can take several hours to connect a data point on a single interaction. And that is why we've moved to doing these solution-based methods via a plate-based assay. And then this technique is called mesoscale discovery solution equilibrium titration, or MSD set. But it does have issues as well, in that it can be quite a tedious process. There's a lot of multiple washing steps and precise contact times, very similar to a plate-based ELISA experiment. And one of the other issues is that in order to really do this in a high throughput manner, you do need some liquid handling automation in order to really make this go. So, if you can't afford a Kinetics and you don't want to buy an MSD instrument or a liquid handling robot, this Forte Bio ELISA assay can be a good alternative for you. So one of the issues we had to overcome was the Forte Bio quantitation sensitivity issue. As many of you Forte Bio users are aware, there are detection limits with the off-the-shelf sensors we get from the manufacturer. Here I show the responses from a typical quantitation standard series. As you can see, the responses are quite weak once you get down into the low nanomolar, single-digit microgram per mil range. This makes it next to impossible to observe the low concentrations we typically need to detect for these solution equilibrium titration experiments. So that became less of an issue when we learned about this Forte Bio application note summarized on this slide. Here was described a technique for the signal enhancement of quantitation samples by adding an ELISA detection step. We used a horseradish peroxidase labeled antibody and colorimetric detection reagents and found that they could measure samples as dilute as 2 nanograms per mil, which translates to concentrations in the low picomolar range. With this knowledge, our scientists here at Adimab set out to develop the Biolayer technique. So in order to convince ourselves that we could see the same sensitivity enhancement, we ran a side-by-side experiment using a high-affinity lysozyme binding antibody. We were able to show that using the streptavidin HRP reagent and a biotinylated antigen, that we would also observe the same a strong signal enhancement, one that's greater than 1,000-fold enhanced. So on the left here, you can see we're down here in the nanomolar scale, and in the inset, things deviate off the standard curve at around 3 nanomolar, whereas over here on the right, in this figure, we're measuring picomolar amounts of quantities, and we're way down into below a single picomolar amount of reagent that we're detecting on the sensor. So one thing to point out here is we are using this Biolayer is technically a quantitation assay, and so we're using the binding rates as data points for this Biolayer technique. And this data has been published in the Forte Bio Winter Interactions newsletter. So if you need any more detail on that, you can go look that up on the Forte Bio website. So the first step in the experiment is to do sample preparation. And so sample preparation is similar to most solution equilibrium titration experiments. The idea here is to keep the antigen concentration constant and vary the antibody concentration. We typically incubate these samples between 18 to 24 hours, and the technique is based on the idea that we can detect free amounts, small amounts of free antigen from an equilibrated mixture. And with the setup, we're able to look at least 8 interactions per plate. So here's the schematic of the assay design. We chose the proteinase sensors for their high capacity to capture antibodies. The idea here is to actually build an antigen capture sensor. So we do this by loading a high density of the antibody binding partner, that's shown here. And then we do come in and we block with an irrelevant antibody in the next step. The idea there is that when we come in with this next step where we're going to actually bind antigen, we're also bringing in some of that antibody in there, and we don't want that antibody to cloud the actual binding event. We just want to really only have antigen binding to the sensor. In the next step, we come in and we label that antigen. In this case, it's biopenylated, and so we use the horseradish peroxidase streptavidin reagent. And then lastly, we come in with the signal detection. And in this case, we use a cocktail of that's from this colorimetric version of the ELISA assay, diaminobenzene with a metal catalyst and a hydrogen peroxide. And essentially what happens is the oxidation step kicks out a very reactive precursor that actually affixes itself to the sensor tip. And so basically the byproduct of this dab oxidation binds directly to the sensor. And it does so in sort of a stoichiometric manner in that the response that you get on your sensor tip is proportional to the amount that's that's proportional to the amount of antigen that's actually captured on the sensor tip. So here's the procedure that we use. So the first step, we're calling it step 0, can be done offline, and this is the step where you are actually building up the sensor tip to become an antigen detecting sensor tip. And so the first step is to capture the antibody of interest and then block it with an irrelevant antibody. The step 1 is what we're calling here is that we capture the free antigen from these antibody-antigen mixtures. You can see, over here in this figure that you don't see very measurable amount of binding, especially relative to what you see when we do the actual signal enhancement. And so that you don't see a whole lot going on the Forte Bio until you get to the end of the experiment. So after step 2, after you've got your antigen on the sensor, the next step is to come in with the horseradish peroxidase streptavidin reagent and label that antigen on the sensor. And then lastly, you come in and you dip that into the signal amplification reagent. That's where you start to see increase in binding, and that's where you start to see that precipitate, in quotes, precipitate, adhere to the sensor tip. You can do washes in between steps 1 and 2, and then steps 2 and 3. If you plan on reusing the plate, that's a good idea. And with this type of an experiment, you can see we're at about 900 seconds, so 15 minutes. And we're doing this on a Forte Bio HTX instrument, so we can look at 96 interactions at one time. That gives us a throughput of about 32 kD determinations per hour, which is a great throughput for this experiment. So as you see, this technique works really well. The raw data from a single antibody antigen titration series looks like the figure shown here on the left. For samples with excess antibody, you see very weak signals, and vice versa for samples with excess antigen. Again, we measure the binding rates using the Forte Bio data analysis software, and then plot those values versus antibody concentration. Fitting the data provides an estimate of the KD. And one thing to note here is that the 50 picomolar antigen concentration that we used in this particular experiment is about near the limit of detection that we recommend for this technique. You can see over here on the left, the 8 nanometers. If you cut that in half and go to 4, you are sort of crunching your signal window and making it fairly narrow. And so, we recommend going no lower than about 50 picomolar. So my last real slide here is to show how this data set compares to what we consider our gold standard assay, the MSD assay. And so for us, it actually compares really well. Here I showed just a small sampling of a larger data set for which we collected both ELISA, ELISA data, and MSD set data. And as you can see, the data look very similar. All the curves look about the same and much better. And even better, the KD values are actually very close to one another, essentially within twofold. And so just a simple ratio shows that, they're within a half or 1.6 of each other, which is definitely within the limits and error bars on this particular experiment. So in conclusion, I think we've shown that BLI-ELISA is an effective tool for measuring low picomolar antibody-antigen affinities. It has the benefit of being more high throughput than the gold standard Kinetics assay and is much less tedious than our in-house gold standard high throughput method, MSD set assay. And what's even better after all of this is that the results from this BLI-ELISA assay compare well with the MSD set experiment. And lastly, I want to acknowledge Yuchi and Patty for their initial work on this assay, Yao for collecting almost all of the data shown near the end of the talk, Yingda for his guidance throughout this entire project, and Dane and Tillman for providing us with one of the best working environments I've ever had in my life, and for allowing us to take on these types of endeavors.
Excellent, Mike. Thanks. You provided our audience with a great application of BLI-ELISA assay and showed us the relevancy to a real-world drug discovery process, so we thank you for that. Before we start our Q&A session, I want to let everyone know that this is your final chance to submit questions for our speakers. So if you haven't done so, hurry up and send them in now. Okay, everyone, so it looks like we've come to our Q&A session now. We've got a lot of questions, and we're probably not going to be able to get to all of them, but we're going to answer as many as we can, and anything that we don't get to, we'll be able to answer offline. And we apologize again for any technical difficulties in the beginning. Sorry about that, but we're back online. Everything's good to go. So our first question is going to be for Dominic. Dominic, one of our audience members would like to know, what are the surface immobilization chemistries for BLI?
Thanks, Jeff. That's a good question to ask. So on my previous slide, I showed the menu of 17 different chemistries or 17 different biosensors. And so there's quite a bit to choose from. The best is actually to be able to check out our website and be able to select a particular biosensor and get more information about the specific chemistry they're using for that particular biosensor.
All right, great. Thanks, Dominic. So our next question is going to be for Vishal. Vishal, one of our audience members would like to know, can Octet do PM binding affinity without labeling in a label-free assay?
Yes, I mean, it's similar to the SPR. You can measure picomolar affinity interactions, and I have cases wherein I've measured characterized 5 to 10 picomolar affinity across the board using the Kinetics, Octet, and the Biacore, and all the values were within threefold of each other. However, if you want to characterize picomolar, you would have to significantly reduce the ligand surface densities. In my case, to characterize that particular interaction, I had to capture around 0.1 to 0.15 nanometer of the antibody, a strategy which you would typically adopt even on the SPR platform.
Okay, great. Thanks, Vishal. Next question is going to be for Mike. Mike, are the free antigen capture step times, comparable between BLI, ELISA, MSD set, and Kinetics?
They're slightly different, Jeff. For the MSD set assay, we typically use about a 2.5-minute antigen capture step. For Kinexa, you can vary that by moving the flow rate through the bead pack from anywhere from like 50 microseconds to about only a second. So that's sort of your best case scenario. And for obviously, just as I spoke about, we use about a 10-minute antigen capture step in the case of the Biolayer, and it's mostly because we want to be able to detect that small amount of free antigen that is covered up mostly by antibody. So that's the basic answer to that question.
All right, thanks, Mike. Dominic, we're going to swing back around to you, and one of our audience would like to know, what is the limit of detection of the Octet platform?
Okay, so realize that we have multiple or different types of Octet systems. And so from a bird's eye perspective, our highest sensitivity systems are the Red 96, the Red 384, and the HTX when it's in 8 or 16 channel mode. And those will measure 150 Daltons as far as molecular weight, and 1 millimolar to 10 picomolar as far as affinity. For folks that are mostly working in large molecule, we have the QKE, the QK384, and also the HTX when it's looking at 36, 48, and 96 channels all at once. And those measured molecular weight levels at 5,000 Daltons, and the affinity range is 0.1 millimolar to 1 nanomolar.
All right, thank you, Dominic, for that. The next question is for Vishal. Vishal, our audience member says, rebinding will be more of a concern for slow off rate. I do not think that your statement of no rebinding will be valid for a slow off rate interaction. Do you agree?
Well, rebinding is always a concern with be it the BLI or SPR, but especially for slow off rates, my biggest concern would be a capture step. So but the strategy would be to directly immobilize the ligand on the surface with low density, and then do the kinetics assay. That would be my recommendation.
All right, thank you very much, Vishal. Mike, this one is for you. Have you compared the results from the BLI ELISA with Kinexa data? If so, how well do they compare?
So in a roundabout way, we have. We've actually measured, we've compared our MSD set data to Kinexa data and shown that we have about a 2-fold comparison between those. And so in a way, by doing this comparison that I just showed just now, the BLI ELISA, it compares about 2-fold as well.
All righty. Dominic, for you, someone would like to know, do you have an explanation for the fact that in some cases I observe inverted sensograms?
Okay, great. Actually, that's nice. That's a good question. It's one of the things I showed on my earlier slide. And when you look at the interference pattern and you look at that delta lambda, or that shift, going from reference to actually dipping into your sample well, you'll see that delta lambda as an example as a small shift. If you have a larger molecule or causing a larger shift, that's when it could go ahead and invert the sensogram. And so we've seen this. We've had people ask about it in the past. So we had something included in our software where the software will actually correct this inversion in the display. And that's something that people have been working with nicely for them. And also, if you just want more help or more explanation, you can certainly talk to our tech service. We've had a lot of people actually ask about this inversion when it comes to looking at virus-like particles, and it's happened with those large types of analytes. And again, that's where we've helped them with this software feature and also talking to our tech support with hot topics like virus-like particles.
All right, thanks, Dominic, for that. Vishal, one of our audience members wants to know, is it necessary to perform buffer reference subtraction on Octet?
Absolutely. Well, in the case of the Biacore instruments or any other SPR, we always do double reference. That would be the ideal case, but if it is extremely important to at least do single buffer reference subtraction. And the whole idea is simple. It's trying to compensate for any bulk shift, even if it is very, very small, to the range of 0.01 nanometer, and it's also to compensate for any observed slight dissociation of the captured antibody from the sensor. So for any kinetics assay, single buffer reference subtraction is a must based on my experience.
All right, thank you, Vish. Mike, one of our audience members asks, how long do you wait to reach equilibrium?
So we typically do overnight, so anywhere from 18 to 24 hours is our typical incubation step. We have gone to 48 hours, but you do tend to compromise the integrity of the interaction. Things can, antigen stability becomes a factor if you start waiting that long. You can go to colder temperatures. So if you were to incubate at 4 degrees instead of at room temperature like we typically do, you might be able to extend that incubation time for super high affinity interaction.
Great, thanks, Mike. Dominic, how good is Octet with small molecules, say 200 to 300 Daltons as analyte?
The Octet is quite good with small molecules. So as I mentioned for the previous question, for the Red 96, the Red 384, and the HTX in the 8 or 16 channel mode, they measured down to 150 Daltons. A lot of people have actually asked about that. Seems like they want to make sure they get their experiments just right, or they're comparing. They want to compare their results maybe as an orthogonal validation to another platform. And like I said, it works quite well. We've helped people optimize their experiments, and people have been able to successfully and reproducibly go down to 150 Daltons.
All right, thanks. Mike, we have one for you. Are the free antigen capture step times comparable between, actually we already asked that one, Mike, I'm sorry. So we have another question here. This may be either for Vish or Mike. Can we regenerate the Pro-A sensor for the BLI ELISA?
That sounds like it's a question for me. And we don't typically do that. The precipitate that forms on there is really hard to strip off the sensor, and so this is a sensor tip throwaway assay. We've tried and attempted to regenerate them, but we haven't had much success.
All right, and I think with that, looks like we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on our site for 6 months at www.genengnews.com. So if you missed parts of it, or again, in the beginning we've had some issues, if you'd like to re-listen to that again, or forward the link to friends or colleagues, which we always recommend. I'd like to thank Dominic, Vishal, and Mike again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to Forte Bio for sponsoring this webinar. So hopefully we'll see you again in another Gen webinar in the near future. And goodbye for now.